All right. So uh, um, our next talk will be Nurul Anwar, and he's not Adriana Zanka, if you might have noticed, and that's because she had to pull out last minute, which is quite unfortunate. But he's very graciously stepped in at the last minute and prepared a talk for us. So hopefully we can all enjoy whatever he has to talk about.
Thanks everyone for being here today. So my name is Anwar. I'm a PhD student at the University of Ireland with School of Mathematical Statistics. I apologize if I'm not the person that you wanted to see. Apology from Adriana. She had to had some emergency stuff, so she couldn't be here in person today. So today I'll be talking about the optimal interruption of the Vivax transmission with the mass drug administration with the help of a multiscale model that we have developed as a part of my PhD project. So as Young already talked about the malaria. So malaria is an infectious disease and it's mostly transmitted by mosquitoes. And according to the WHO, so 241 million of malaria cases were only found in 2020. And out of these cases, there are 627 of death because of these malaria parasites. There are 5 species of the malaria parasite that causes the disease, and out of them, the Plasmodium vivax is currently the most globally widespread parasite. And most of the Vivax or the malaria cases are happening in the African region, where 95% of the total global cases are happening. And out of those 95% of the cases, 75% of cases are happening because of the Vivax parasite. So the question is, why the Vivax has become the globally widespread parasite? So to understand that, we have to look at particular part of the parasite's life cycle. So whenever an infected mosquitoes bites an susceptible human to take bloods, so the parasite in form of sporozoites find their way into the human body, and then it found their way into the human liver. While being in the liver, some sporozoites start to develop into other forms, which develops into schizoid, and then merozoites. These merozoites are then released into the human bloodstream. So whenever these merozoites are found in the human blood, so the symptoms may appear, and we say that the individual is blood stage infected. So the significant characteristics of this Vivax parasite is that some sporozoite can still remain within liver as dormant. We say that hypnozoites, and these hypnozoites can activate after a week, after a month, or even after a year, and gives rise to they goes through the same development process and gives another blastose infection. So whenever the parasite are found in the liver, or say hypnozoites, we call that the individual is liver stage infected. So this is why it's become the most globally widespread one. And it's believed that almost 80% of the total Vivax cases are because of this hypnozoites activation. That means they can activate, and we say that relapse, and they go through the same, blastose infection, the symptoms, and the same. So usually we use the Ross-Macdonald framework to model the mosquito-borne diseases. So I believe you already have some idea, but for the sake of my talk, I'll just go through this. So this is the deterministic compartmental approach. So we divide the human population into certain groups. In here, fraction of susceptible for as, fraction of infected human, and fraction of recovered human, and as well as the mosquito population. And we use the differential equation to track down the rate of change in between this compartment. And these are actually a nonlinear ordinary differential equation. So there is no analytic solution. So we use a numerical approach to find the solution. So these Ross-Macdonald framework is best describes the other parasite, which is the Plasmodium falciparum. But since the Vivax is significantly different because they have another stage, which is the hypnozoites. So these model cannot be used to describe the model dynamics of the Vivax one. So because if we wanted to capture the proper dynamics for the Vivax life cycle, because if you get bitten by mosquitoes, you might have different number of hypnozoites established within your liver, right? So if you have 5 hypnozoites, for example, 5 hypnozoites established after you get bitten by an infected mosquitoes. So currently you are, say, infected, but you have 5 hypnozoites. So if the hypnozoites die because of the death of the host hepatocyte cell, so you might have one less hypnozoites. That means currently you have 4 hypnozoites, but you're still infected. But if you recover from an infected or infected status, you become susceptible, but you have still hypnozoites. That means you are susceptible to have relapse and again suffer from blood stage infection. So that's like, so if you are susceptible, you have 2 hypnozoites, and if the hypnozoites activate, then you have 1 less hypnozoites, but you're still infected. So that's the complicacy of the Vivax different disease state. If you wanted to capture the more realistic and as well as capture the hypnozoites within individuals. So our main idea is to, so why we wanted to create a model, right? Because there are so many Vivax model in the literature, but most of the model only considers one compartment to represents the states of having hypnozoites. So it's like just considering that individual have 1 hypnozoites, but that's not true. Individuals might have different number of hypnozoites from different, mosquito bite. Sorry.
When an individual gets infected, how many hypnozoites?
That actually varies because some literature, one of the model which is developed by White, they estimated that on average it's 8.5. Oh. So that's why, like, so it's not accurate if you just consider that individual have hypnozoites. So if you wanted to capture the realistic dynamics, we have to consider the variation of hypnozoites within individuals. So that's how our aim comes in. So our primary aim is to develop a population model for the transmission dynamics, and then embed an within host model to capture the effect of the hypnozoite dynamics into the population scale. And then study the effect of the radical cure treatment with the method of mass drug administration. I'll come talk about this later. And then the optimal interval study. So we use the deterministic approach. So we use 3 compartments represent the fraction of susceptible, fraction of infected. So whenever I say infected, that means individuals have parasite within their blood, and H to represent the fraction of liver stage infected individuals. So just to remind you, the liver stage infected individuals have at least 1 hypnozoites within their liver. But susceptible individuals do not have any hypnozoites within their liver, but infected individuals might have or may not have hypnozoites within their liver. So they might vary. So some might have no hypnozoite, but some might have different number of hypnozoites. So susceptible individuals can become infected if they get bitten by an infected mosquitoes. So while they suffer or while they're in the infected compartment, if they recover, so that gives you 2 different scenarios. So they can either move or become susceptible, or they can become liver stage infected. So that's why we have defined a parameter PT, which is the probability that an infected individuals have no hypnozoites within their liver. So if they don't have any hypnozoites within the liver, after recovery, they become susceptible again. But if they have hypnozoites, then they become the liver stage infected. And while they are in the liver stage infected compartment, so if the hypnozoite activate, they become blood stage infected again. So we have defined a parameter KT, which represents the average size of the hypnozoites reservoir, given that they are liver stage infected individuals. And we have defined alpha and omega as a the constant parameter, where alpha is the activation rate of the hypnozoites and omega is the death rate of hypnozoites. So liver stage infected individuals can still become susceptible if they had one hypnozoite and that hypnozoite died before they get the chance of activation, right? So then they can become susceptible again. So just a while ago, I said that we wanted to properly capture the realistic dynamics for the Vivax because they have different number of hypnozoites in the liver. So how can we capture the variation of hypnozoites within individuals with just 3 compartments? So this is where the within host model comes in, because the parameter P's, K1 and KT, they're actually derived from the within host models. So the within host model is developed by Soumya Mehra, and this is a special case for short latency. So whenever I say short latency, it means that hypnozoite can activate immediately after establishment. But for long latency, they go through some dormancy stage. That means they cannot activate before they have some waiting period. So, but this is for the tropical strain, which is the short latency case. So the first, they model the dynamics of a single hypnozoite. So a single hypnozoite in the liver can either activate at a constant rate alpha that gives rise to a blood stage infection, or they can die at a constant rate omega. But after they activate, these blood stage parasites are then cleared with a rate R. So they have 2 final states. So they can either be removed by death or by activation and through clearance. So the model uses the Kolmogorov differential equations to track down the probability of that hypnozoites of being each of the different states. So then the model a single mosquito bite, assuming that a single mosquito bite contributes to some number of hypnozoites, and then we allow continuous mosquito bite throughout the simulation time. So in order to consider continuous mosquito bite, we assume that the hypnozoite dynamics are independent and identically distributed. That means each of the hypnozoites act independently. Doesn't matter. It's like so in literature, one model which is developed by White, they assume like a collective dormancy. That means if you have a batch of mosquito bite from a mosquito bite, so like if you get bitten by mosquitoes and you have say 7 hypnozoites, so their assumption was that 7 hypnozoites process through the same rate. That means if activate, the 7 hypnozoites activate together. But this model, it actually assumes that each hypnozoites act independently. That means it doesn't depend, doesn't matter when they're established or which from which bite. And we also assume that the mosquito bite are Poisson distribution with the time dependent with lambda. And we assume that the number of hypnozoites per mosquito bite is geometrically distributed with an average number of hypnozoites given by mu. So with that, we can create a probability generating function that gives us the number of hypnozoites in different states. So how can we use these probability generating functions to obtain our within host parameters that we need in our population scale model? So just to give you an example, so if we wanted to create or obtain that the probability that individuals have no hypnozoites within their liver, they are not suffering from any relapses. So N is like the number of activation or the relapses, and they don't have any primary infection as well. So this is just to substitute the values in the probability generating function, and then we can get the probability. So from here we combine the 2 models together. So how the couple model works. So usually for convention model, infected mosquito bite human becomes infected, and also if a susceptible mosquito bite an infected human, that susceptible mosquitoes become infected. But in these cases, our model actually can track down all of the mosquito bite that an individual have received throughout the time, and gives us the probability that what's the probability that individual will have 0 hypnozoites, individual have 1 hypnozoites, and so on. So this is how our say couple model work. So we can solve the model numerically, and just like any other model, we can have like the dynamics of the transmission. So we can have like the idea how many proportion of individuals are blood stage infected. So blood stage means they have blood stage parasite in the blood, and how many proportions are individuals are liver stage infected. So in addition to like the convention result, our model can provide like the hypnozoite distribution. That means what is the probability of having different number of hypnozoites within the liver. So on the top right is the overall hypnozoite distribution. The bottom left shows the distribution given that they are liver stage. That means the individuals in the H compartment. And the bottom right is the distribution in the I compartment. So we also run a sensitivity analysis over for different key parameters, which are some number of mosquitoes, number of average hypnozoites per mosquito bite, the hypnozoite activation rate, and hypnozoite death rate. So for mosquito number, which is an average hypnozoite size or per bite. So if the both increases, that means if there's too many mosquitoes in a region, that means individuals will become, more individuals will become blood infected, right? So that's why the increase monotonically and register 1. But for liver stage infected proportion, so we usually know if they have too many mosquitoes, that means there is a more chance of getting more hypnozoites in the liver, right? So why there is the hypnozoite liver stage fractions decreases, since we are assuming that each mosquito bite gives rise to a blood stage infection. So most of the individuals are most probably are suffering from a blood stage infection, even though they have hypnozoites, because in our model we assume that infected individuals may or may not have hypnozoites within the liver. I'm not going to talk about all the details, but if we look at particularly say for the activation rate alpha, so when the activation rate is 0, that means hypnozoites have only one chance of dying, right? So they cannot activate. That means the disease cannot progress, so it goes to disease-free equilibrium states. But as the activation rate starts to increase, so there's the relapse are happening. So we are seeing increases in prevalence or increases in blasters infections. So when the activation rate is very slow, the hypnozoite reservoir started to build up. But as the activation rate increases, that means there are more, the relapse are happening more frequently. So it's like the build up hypnozoite reservoir are decreasing in size. So even though when the activation rate is too high, there is not enough blood infected fractions, because as I mentioned, there is no enough or not enough reservoir hypnozoites in the reservoir to continue the dynamics. So then we applied the treatment in our model. So in the method of mass drug administration. So for the radical cure is a combination of 2 drugs. As I mentioned, said that the Vivax has the 2 different parasites. So one is in the blood, and one parasite, which is the active, the hypnozoites, they stay in the liver. So the 2 drugs, one drug, which is the combination artemisinin combination therapy, they can kill the blood cyst parasite, and the primaquine or tefenaquine, they can kill the hypnozoites from the liver. So the radical cure is a combination of these 2 drugs. And these are given in the method of mass drug administration. So the mass drug, so conventional way of treating individual or giving drug to individuals, like we have to screen individuals who are infected and then give them drug. But the mass drug administration is like give the drugs to the whole community regardless of infection status. This is like if the screening process is too complicated, because it's very hard to detect the hypnozoites, because currently it's been proven that not only liver, sometimes the spine and even in the bone marrow, the hypnozoites can stay in the bone marrow as well. So it's very hard to detect the individuals who have, say, hypnozoites within the liver. So that's why the method is like give the drugs to the whole individual, in a whole community, regardless of infection status. But this has some limitations because the drug which kills the hypnozoites, the primaquine or tefenaquine, they have some side effects. So they cannot be prescribed to individuals who have glucose 6-phosphate dehydrogenase deficiency, because they can cause severe blood hemolysis. And also they cannot be prescribed to pregnant women, because that also makes things complicated. But currently in our model, we haven't considered this side effect yet. So we assume that everyone is given the drugs. And we also assume that the drug has an instantaneous effect. That means when the drug is applied, the ACT or the combination of the artemisinin drug, they can kill the blood stage parasite instantaneously. So we assume two parameters, which is P blood, the probability that the ongoing blood stage infection is being cured at instant. And also we defined a parameter which is P rad, that gives the probability that the hypnozoites die instantaneously. So in addition to the hypnozoite death rate, which is omega, now there is an additional rate that the hypnozoites die, and as well as any ongoing relapse or any ongoing infections are being cleared, in addition to the normal recovery, which is R, are being cleared with the red, the blood. So now we want to see the effect of the radical cure or the MDA in our population scale model. So the top two shows like without the treatment. So, and we are also seeing the hypnozoite distribution at steady state. But if we apply the MDA, so when the system is in steady state, we are seeing like the decreases or the effect of the drugs in the proportion of blood infected individuals. But as you can see, for the liver stage infected individuals, there's a slight increases when the drugs is applied. So this is due to the effect of the efficacy of the drug. As currently we're assuming that the radical cure drug that targets the hypnozoites, they are not 100% effective. That means they cannot kill all the hypnozoites. So in model, we mentioned that those individuals who have developed hypnozoites after recovery, they become liver stage infected individuals. So since the blood stage infected fraction is decreasing, that means they don't have any blood stage or blood stage parasite right at the moment. So that means they now have the chance of become susceptible again or become liver stage again. But since the radical cure drug cannot kill cannot kill the hypnozoites 100% effectively, that means individual will have still some hypnozoites within their liver. So that's why there's a slight increase in the liver stage infected compartment. And if you look at the hypnozoite distribution when the drug is applied, we can see that the radical cure, effectively can reduce the hypnozoite reservoir size as well. But if we look at the long-term behavior after the treatment was applied, it looks like there's no effect at all. This only has the effect in the short time period. And you all can also check it by looking at the distribution before the radical cure was applied and say after 5 years the radical cure. Because since we're assuming that individuals are constantly receiving mosquito bite, and the mosquito bite contributes to blood stage infection, and from each mosquito bite, you are having some proportion of, or on average, new number of hypnozoites. So, and hypnozoites can activate and gives you relapse. So it's like the system becomes, same again. So it's like there is no long-term effect. So then we wanted to check what happens if we apply multiple MDA with radical cure. So as you can see, so this is just to give you examples. So 3 MDA when the system has, it's a 50% prevalence. And we can see that like if we're applying more MDA, that means we can use the prevalence more, right? So then we run a sensitivity analysis over 20 MDAs over 20 different statistic prevalence. So it's like when they did from 20% to around 85% for 20 different MDA. as you can see, for initial a number of, say, up until 8 or 9 number of MDA, the prevalence decreases, but then they increases. So why is that? So we wanted to have a look with the time series. Just to should give you an idea. So these the heat map shows you the minimum number of prevalence after each MDA. So as you can see, so this is the minimum MDA after minimum prevalence after 2 MDA, and this is the minimum prevalence after 3 MDA. So this is the overall minimum prevalence after 20 MDAs for different statistic prevalence. So if we look at the time series plot for that particular, say 50%, when the prevalence is 50%, as you can see, say when, and also I wanted to mention that these MDAs are given at a 2-month interval. So after 2 months, the second MDA, and then after the 2 months, the third MDA. But if we look at the time series, the prevalence, say after around say 6th or 7th MDA, the prevalence doesn't decrease at all. It's like going up. So why is that? If we look at the average hypnozoite reservoir size over the time period, as you can see that the radical cure can reduce the hypnozoite reservoir size, but since individuals are still getting mosquito bite, and before the next 2 months, the hypnozoites started to build up more than it decreases the size. So effectively, the radical cure doesn't have much more effect. So that's why for like the next rounds of MDA, the prevalence already up. So it cannot bring down the prevalence more. So then the question comes in, what happens if we apply the MDA? Yeah, so, sorry. Yeah. So then we look at like the frequency within 1 month interval. So this obviously works better compared to 2 months interval. And if you also look at like the side-by-side comparison, if we apply the radical cure or the MDA with 1 month interval, they are more effective compared to 2 month interval. So that means the interval plays a significant role in this here. So then we model an optimization problem. So our idea is to, or our approach or objective function is to minimize the prevalence with the given interval. And I don't have enough all the results so far now, but as you can see, we're seeing that the optimal interval has the more effect compared to other fixed time interval. And if you look at the time series plot, as you can see, the overall prevalence is decreasing in trend. But if you look at the interval, so instead of fixed time interval, now we have variable interval time. So as the number of MDAs are increasing, like the MDA have to be more frequent. So yeah, and this also significantly can reduce the size of the hypnozoite reservoir, right? So if you see the overall effect of the optimal interval, so optimal interval is the more effective compared to having fixed term MDA and interval. But no matter which way you see, the MDA has only effect on like short-term interval, but in the long run, the drugs does not have any effect, right? So it's like MDA alone is not sufficient enough to eliminate or eradicate malaria. So yeah, like in short, I talked about how we developed a population model and how we have embedded the within host model into our population scale model, and how we have coupled the model to consider treatment with the method of mass drug administration. So in future, our idea is to consider other sort of treatment strategy in addition to the mass drug administration. So like the vector control. Yeah, with that, I'd like to thank my supervisor, Jennifer Flagg, all my supervisors, and a special thanks to Samia for the within host model. Thanks everyone.